So thank you, Alana and Ryan. That was great, and I think it's really nice to see visibility into what we're doing, why we're here, and understanding we're a scientific organization that is our primary and only mission is to advance science to improve the lives of our loved ones and our children living with Angelman syndrome. And so it takes a lot to do that. And we got to the point where the science has been so promising and the growth has been exponential that we couldn't do this on the backs of caregivers volunteering before work, after dinner, at night when the kids go to bed, weekends, birthday parties, and Christmas. It just wasn't possible. I couldn't do it anymore. So it allowed us to bring in an amazing team to do it right, to do it faster, to do it safer, and to ensure that we're as thorough as we can, because that's what our kids deserve. So this is a really huge time for us, and I'm so proud of what you guys have done, because I didn't have the courage to do it. So to Alana and Ryan, thank you. So now let's talk about the roadmap to get there, because we're not there yet, and that is the reality, but I'm excited at how we are accelerating. And so I'm going to really talk about the roadmap to a cure and how we're accelerating drug development for Angelman syndrome, because it's pretty profound. And I don't want anyone in the room to be scared to ask a question. We are here for the next 2 days for parents and caregivers. That's why we're here. We're here for you to ask every question you want that you've been waiting for, that wasn't answered on social media, that wasn't answered by your colleagues, your peers. We're here to answer them. Our researchers are here to answer them. The pharmaceutical companies and the biotechs are here to answer them. They are here to give you the answers that you've been waiting for if they can, but also be prepared that some questions are a bit hard to answer, and you may not be able to get an answer today, but we're working on ensuring we get all of the answers. So as most of you know, I already told you I'm the Chief Science Officer here at FAST. I'm also the co-director of something called the Angelman Syndrome Biomarker and Outcome Measure Consortium. We had an incredible day yesterday. We had an entire day from 8:30 to 6 with, I think it was about 35 talks, maybe 40 talks in one day, which is masochism. But it was a day of researchers, scientists, and clinicians all getting together to talk about science in a deep way in which we could share information. And I'm gonna have another talk later this afternoon just to go over with you what the ABOM is. You hear a lot about that term, the Biomarker and Outcome Measure Consortium. What does that mean? Well, a lot of our money has gone into that, and I think you deserve to really understand the output of that and what it's all about, because it's really important. It's a little less sexy than seeing a gene therapy in the brain of a mouse, and I'm going to show you a lot of that today, and you're going to hear a lot of that from the other talks today and tomorrow. But it is something that's critical, and it's where a lot of our resources go, and it's where a huge effort that FAST and the ASF have put in. So I'm going to share that with you later and my role there. But I'm also the co-director of something called the International Angelman Syndrome Research Council, which I'll explain in a couple of slides. I took on a new role this year as the Chief Development Officer of our accelerator called AS2 Bio, and I'm going to talk to you a little bit about that. And by training, I'm a veterinarian. I'm a dog and cat doctor, that's what I am. I develop medical devices for animals with naturally occurring disease, and I've been running clinical trials for a long time, proving if these devices work when you have a disease that's not fixable, yet if we can develop a device to fix it, then it's a way to solve a problem. So it served me well when Quincy got diagnosed with Angelman syndrome, because she was broken and we needed to fix her. And I came from a background of fixing things that didn't have a solution. And I felt at the time, Angelman didn't have a solution. How can we fix this? Which brings us to the most important job that I have, which is that I'm the mother to 3 beautiful little girls. Their names are Kai, Quincy, and Piper. And Quincy, my middle daughter, lives with Angelman syndrome, and she's pictured here on the right. So in disclosure, I'm the co-founder and former chief operating officer of a company called Genetics Biotherapeutics that they just referred to, and that was acquired in 2022 by Ultragenyx Pharmaceuticals, and I still serve as a consultant to Ultragenyx. I'm a strategic advisor to other biotech companies that work in neurodevelopmental rare genetic conditions, one of which is called Mosaicism Therapeutics. I serve on the External Scientific Advisory Board for Biogen in their drug development division, and I'm a consultant and I advise many different initiatives in rare disease drug development, particularly related to Angelman syndrome. So I work with every pharmaceutical company in the room, doing everything we can to help them safely and quickly advance their therapies toward treatments for Angelman syndrome. So let's get down to the science. This is the most important part, because as you all know, knowledge is power. And if you understand Angelman syndrome and the genetics behind Angelman syndrome, that is the only way you can be well-versed to realize and understand how we have the potential to fix it. And you are going to need to be making very important decisions about your loved one living with Angelman syndrome. What's going to be the best for you? What's going to be the best for your family? What are the risks? What are the benefits? How is this delivered? What's a virus? How does this go in? Does this carry a virus? Does this not carry a virus? All of these things are critically important. So I'm going to walk you through a little bit of science behind Angelman syndrome, because I think if you understand this, you're going to really understand so much. We have a glossary of terms that should be in your bags, or they're also online, where we're going to talk about a lot of terms in the next 2 days. And you don't need to remember everything. There are a lot of acronyms and a lot of terms. What I want you to do is I want you to write them down and then look in the glossary. And anything you don't understand, I want you to come up to me or any scientist in the room and ask the question. What is this? Explain this to me and explain how this is relevant to my situation and my child. Because again, you need to understand everything in the next 2 days for you to be able to make the best decisions and for you to be able to have the hope that I have. And the reason I have it is because I understand it. And so I want that for you more than anything. So we all have in almost every cell of our body 23 pairs of chromosomes. We get 1 copy from mom and 1 copy from dad. That leads to 46 chromosomes per cell. And you can see this picture, this karyotype on the right. And that karyotype aligns the chromosomes 1 through 23. And because of that, each one of those chromosomes carries DNA. And DNA together create genes, and genes ultimately make proteins. And proteins are how our body function, by having the right number in the right spot that are expressing at the right levels. UBE3A is a gene. It is on chromosome 15, and it functions on both the maternal and the paternal allele. But I'm going to explain to you how that's a little bit different in the brain. The UBE3 gene is what is impacted in Angelman syndrome, and generally it's either deleted, mutated, or silenced in individuals living with Angelman syndrome on that maternal or mother's allele or chromosome. 1 in 15,000 individuals are estimated to live with Angelman syndrome around the world, which could translate into 500,000 or more individuals worldwide. We just haven't found them all, and we're working very hard to try to find them. It is random, and it is rarely inherited, though there are situations in which it is inherited, but that is not very common, which means that most of us grabbed a lottery ticket. Wasn't the best lottery ticket, but it was a lottery ticket, 1 in 15,000. And we had that happen randomly, which means you're not a carrier, and likely you will not have another child with Angelman syndrome. Well, you have a 1 in 15,000 chance of having another child with Angelman syndrome. So it doesn't mean you shouldn't have more children. And we have a genetic counselor who can, Nikki Armstrong, who's here, who can talk to you about that, because it's a big question families have. Like I said, every chromosome is made up of genes. Every gene codes for proteins, and those proteins have function. And so let's look at this schematically. Here's chromosome 15. There's 1 copy from mom or maternal. There's 1 copy from dad or paternal. In all of our cells, us as caregivers that don't live with Angelman syndrome, it is read on the maternal allele and the paternal allele. But in all of us, in our neurons, so in the cells of the brain, it is silenced. It is silenced by a natural phenomenon called imprinting, in which there's this long stretch of RNA, which is a type of genetic material, that ultimately sits over that DNA and says, stop. And that's true for me, and it's true for you. And it's true for your child living with Angelman syndrome. But for me and for you, we are reading that allele on the maternal side in neurons. Therefore, we only need one copy in neurons in order to have normal neuronal function. But in our kids, it's missing. It's deleted, mutated, or silent. And because of that, they don't have an expressing UBE3A protein coming from the maternal allele, and it's silent naturally on the paternal allele, which means the neurons have none, where you and I have one. And that results in the symptoms of Angelman syndrome, which is why most of our kids only have a neurologic condition. Peripherally, the cells of their body, the other organs of their body are quite healthy because they have the paternal copy, the father's copy, that is not silenced and is being read, which is why we target the neurons of the brain in order to try and give back this gene. And we can give that back in many ways. So, like I said, there's many different ways or genotypes that you can have Angelman syndrome. One is a deletion. About 70% or so of our kiddos have a deletion where a segment of the DNA is missing on the mother's copy. Some have point mutations. About 10 to 12% have one letter that is abnormal. It was written wrong, so the protein is not being made. About 3 to 6% have double silencing. So like the father's copy is silent, while the mother's copy is also silent. And that's called uniparental disomy or imprinting center defect. They both have a silencing phenomenon for different reasons, but they both have a silencing phenomenon. And then there's something called mosaicism, which to me is really a secret look into what gene therapy can look like. Because mosaicism means that our kiddos with deletions and mutations had this happen at the level of the egg, meaning that every cell made after fertilization, egg, sperm, has a maternal defect. Mosaicism happens after fertilization. So you have a healthy egg and you have a healthy sperm. You fertilize, and then something happens in development in which one of those cells loses maternal expression. Therefore, some cells don't have maternal expression, but some cells that grew before that event happened do have maternal expression. So it becomes more of a mosaic, where you have some healthy and some unhealthy cells, and those individuals have UBE3A and some cells that are expressed, and we're going to talk about that. Well, what's important about UBE3A? Well, we live with what's important about UBE3A, or we live without it. UBE3A is an enzyme. It's a protein that targets other proteins and essentially kind of polices the brain in a way in which it helps regulate how we turn other neurons on or how we turn them off, how the communication happens between neurons. And so it targets proteins for being broken down or being upregulated. It tags them to tell the brain, we're going to knock this down or we're going to upregulate this protein. And it's required for the normal function between neurons. So we have neurons, as you can see here on the left, and they talk to each other. In order for our legs to work, our brain has to tell our leg to work. And that results from the communication from your brain to your spinal cord and from your spinal cord to your legs or to the muscles of your body. And so in order for you to function and coordinate your neurons appropriately, you need to have normal synaptic or junctional function and communication between the neurons in order for them to work well. If there's a deficiency or an abnormality at that junction, it is an abnormality to the synapse. And that synapse, if there's a defect there, is called a synaptopathy or an abnormality to the synapse. And ultimately, overwhelmingly what happens is that we lose what we call tonic inhibition. What does tonic inhibition mean? If I am going to walk, and I talk about this a lot, and this is what makes sense to me and hopefully to you, if I'm going to walk and my brain tells my neuron, I want to walk, right? My neurons are going to communicate so that they can start to activate my muscle and I can take a step. One of my legs is going to activate while the other relaxes, then the other is going to activate while the other relaxes. And you have this combination of activation, inhibition. As I'm talking to you, there are thousands of muscles that are activating and inhibiting in order for me to coordinate my mouth, my tongue, my throat, and my voice box to be able to speak. If you don't have appropriate coordination of the neurons talking to each other and them talking to your muscles, it's really hard to function. Well, welcome to our kids' world. This is their problem. They have so much activity and not enough calming or inhibition that they're always on. And this is why they have challenges with balance. They have challenges with sleeping. They're overactive, so they have seizures. They have challenges coordinating their muscles, so they can't talk. These are all reasons that our kids have the symptoms they have, this lack of coordination. And it's very important to understand that, because if we can coordinate at the synapse better, that would lead to potentially better neuronal function. And that's what our kids are looking for. Now, I want to highlight a little bit more than just UBE3A, because individuals living with the deletion genotype, which is a majority of our population, potentially 70 to 80%, maybe 60 to 70% as we learn more about the incidence, we learn more about the percentages. But what I told you earlier was that they have a large segment of DNA that's missing. A point mutation has one base pair of DNA that's abnormal, usually one, maybe three. A deletion has 5 to 6 million base pairs of DNA that are missing. We wonder why deletion looks different than mutation. Well, they're missing 6 million things that mutations are not missing. The fact that they're not more different is a phenomenon to me that I don't actually understand. If one base pair can make Angelman syndrome, 6 million only makes it a little bit worse. So our kids should be applauded as deletion kids of what they can do, because they have been faced with such an enormous challenge of 6 million base pairs of DNA that's missing. And so this is something we have to understand. And the reason is, what is that difference in their behaviors and their presentation? Is the fact that they are missing a segment of DNA, and if we blow that up, there's UBE3A. There's the entire segment that most of them are missing. And those are the genes that are active on the mother's copy that are missing. But of those genes, not all of them have shown to be associated with disease. So we have many genes that are abnormal that aren't associated with disease. So if these genes are missing, which ones are associated with disease? Because we may need to give those back as well if we're going to truly fix the deletion genotype. And so what we know is on that side, there's one gene that may have impact. On this side, there's 5 genes that may have impact. But when we look into the human phenotype of these disorders, what really stands out are the GABA genes. Those GABA genes probably have the most impact on why our kids with deletions look different. Well, we narrowed it down to what we think we really need to address to make a difference. And if we look at the function of those genes and you really dig into the human phenotype, the human presentation of those genes individually, what we know is that of those genes, the 2 GABA genes are the ones associated with the symptoms that probably increase the severity. So we've narrowed it down from 11 genes to maybe 2 that are actually adding to the phenotype. And this is very important work that hasn't yet been done for Angelman syndrome. And you're going to hear some talks today about the value of that. We have Dr. Levine, who's going to talk about that on a neuron level, and we have Dr. Nadavat, who's going to talk about that on a therapeutic level. So what are the symptoms of UBE3A being missing? Well, I don't need to tell you guys, but I'm going to tell the people in the room that are developing our drugs that don't live with it, because it's important. Our kids have a universal lack of speech. Most kids, 95%, don't speak a single word, ever, in their whole life. Some kids sleep 2 or 3 hours a night. Raise your hand. When they come to an event like this is a vacation, because they actually got to sleep, not only in a bed, for maybe more than 3 hours, but with maybe their spouse, which often doesn't happen in Angelman syndrome. Huge for us, huge. They can have life-threatening and debilitating seizures. I already told you about incoordination. They can have significant constipation. They can be very impulsive and self-directed. Raise your hand if you live with that. Things in the mouth all the time. We all live with that, right? These are all the symptoms of Angelman syndrome that matter and are meaningful to our families. They're unable to live an independent life, regardless of genotype, and that's a simple fact. They can't live alone because they don't have the activities of daily living to be able to function independently. And there's nothing more we all want for our kids than to do that. But let's not take for granted the impact that has on every one of you, family members, who care for our kids. The impact on families should not be underestimated. The lack of sleep, fatigue, stress, anxiety, social isolation, the inability to maintain employment, and the mere cost of therapy, of devices, of equipment, of a bed. Did you guys know that a safety bed costs like $10,000? And insurance often doesn't cover it. Well, thank you to the Angelman Syndrome Foundation, who helps to cover the cost of these supports for our families, because we can't afford it. And this is what it costs to live with a disorder like Angelman syndrome. And we're so grateful for the support that we have around us. And so I talked to you a little bit about mosaicism and why this was a window into possibility. And the reason I said that is because there have been some publications on kids with mosaicism. And those kids generally have between about 1 and 20% of maternal UBE3A being expressed in their blood cells. And we assume that that's about the amount that's in their brain. We don't know that for sure, but there is a correlation generally to the amount of UBE3A expression that we can detect in blood to the severity of their symptoms. So if you look at that graph, if you look down to the right, that's about 1 to 5% of UBE3A. And if you look up to the left, it goes up to about 40% of UBE3A. And then it just jumps to 100%. And that's probably because over 40% is generally neurotypical. They're not documented, they're not diagnosed because they actually have full function. And so if you look at the symptoms of an individual with mosaicism of 1 to 5%, they generally have no seizures, they may have some gait disturbance, and they can speak. If you look at an individual with 10 to 20%, they can speak in sentences. They have no seizures and they walk normally. And if you look at over 40%, they're likely neurotypical. What does this tell you? This tells you you don't need a lot to go a really long way. And if we can give our kids 5%, 10%, or 20% of UBE3A expression, I assure you that is a different child than was born into my family. That is a different child. And I would do anything for that. So we are so grateful to the Mosaic kids who have taught us so much. If we can give 10% to kids with 0 and 10% more to kids with mosaicism, these kids could have the potential to live a different life. So we don't need 100%. And when scientists and researchers talk about not getting to every neuron, not getting to every brain region, not being splattered and scattered, that still could give us this. The enemy of good is better. And one day we want to get to every single neuron. But we're not going to wait 25 years to get there when our kids need something today. And that's where that sense of urgency and reality meets scientific perfection. And we have to realize that there has to be an in the middle. And there will be versions of 1.0 and 2.0 that may give us more and more expression over time. But we're in a place today where we can change lives. And it's time to do that, and I think we are. I think we are. And so I'm gonna go a little bit more quickly now, because now that you understand the science, we can jet through all of the things that are happening in the scientific space. And we break this down into 4 pillars, which makes it really easy to understand. Pillar 1, we can replace mom's gene. Okay, or her protein. So we're gonna replace mom's UBE3A. Pillar 2, we're gonna turn on dad's UBE3A, unsilence. Pillar 3, we're gonna work at the synapse, at the junction between the 2 neurons, and not necessarily work on UBE3A directly, but work on it indirectly. And then pillar 4, are all the tools that we need to do to help accelerate drug development, which is less sexy, but equally important, because we cannot get drugs to patients if we don't understand who patients are, where they are, and how we can measure change that the therapies are actually working. So those are hugely important things. So let's start with pillar 1. Okay, 2 neurons are talking to each other. In the center of that neuron, we have the maternal and the paternal allele, and the maternal allele is missing UBE3A, and the paternal allele is silent from UBE3A. We can replace the gene in the neuron. We can unsilence the gene in the neuron. We can work downstream, and we need to be prepared. Pillar 1, 2, and 3. I like to look at it like this because scientifically I like to look at the neuron and know where we're acting, okay? So now, gene replacement therapy, what is that? Ed's going to talk a little bit more about Therapeutics 101, so I'm not going to go into the details of what's a gene, what's a protein, and what's a virus. But what I can tell you is that with gene replacement therapy, we can actually take a viral vector, that's all the bad parts have been taken out of it, and cargo, the UBE3A gene, has been put into it. And we're going to hear Dr. Wilson talk about this tomorrow for an AAV gene replacement therapy. You can do that with an AAV, an adeno-associated virus vector, or you can do that with lentivirus, where you can actually transform your bone marrow cells, put UBE3A into your bone marrow cells, which become their own factory of UBE3A. Those bone marrow cells cross the blood-brain barrier, get into the brain, and then they start secreting the UBE3A protein. And then the neurons can take those up and function more normally. So that's called hematopoietic blood-derived stem cell bone marrow gene replacement therapy. So you're going to hear AAV gene replacement therapy, HSC gene replacement therapy. That's what we're going to talk about this weekend. And Chris Luthers is going to be talking to us this morning about the HSC gene replacement therapy. We can unsilence the paternal allele, right? How do we do that? We're going to knock down that silencing mechanism. We're going to knock down that RNA. We can do that through an antisense oligonucleotide. Well, we all know a lot about antisense oligonucleotides because those have been in the field for a long, long time in Angelman. We first started talking about those for Angelman syndrome in 2013, 2014. And we have clinical trials for this in humans, which we're going to hear a lot about tomorrow. So we have programs at ASOs by Ionis and Ultragenyx, and Roche is going to talk about their program as well. We have artificial transcription factors, CRISPR gene editing. We can permanently knock down that antisense transcript with CRISPR gene editing. And we have miRNA, which is a permanent way that you can unsilence it by delivering a virus and letting that RNA continue to pump out like a chronic ASO over time and knock it down. And we're going to hear tomorrow from Encoded Therapeutics about that. And then there's the downstream targets. The downstream targets are how can we address the synapse more symptomatically? And we're going to hear from Neurin and Roche about their approaches to those downstream targets, not addressing UBE3A directly, but addressing the symptoms and the consequences of missing UBE3A or the other GABA genes. Okay, gene replacement therapy and enzyme replacement therapy, we already just went over that. Another program we're going to talk about in terms of a maternal gene activation approach is where we can activate those other GABA genes that I was just talking to you about. What if we overexpress the ones that are there, not the ones that are missing, and then we can give back more of those GABA genes? So we're going to talk about that. This is just data, and this is small, and you can't see it. And I don't want to walk you through all the details of it. What I want you to recognize is, firstly, how much work has been done in this space. We have AAV gene therapy that was done out of University of Pennsylvania, University of South Florida, University of North Carolina, Chapel Hill. A lot of people are focusing on how do we replace this missing gene. We have HSC gene therapy, which I just mentioned, that hematopoietic stem cell therapy that started with our program at UC Davis and has now moved to UCLA. What I want you to recognize are the colors on the screen. If you look at the top left, you can see that there's a lot of brown. And then on the small one on the right, there's a little bit of brown. A lot of brown means we went from no UBE3A expression to a lot of UBE3A expression. You could see that we have animals that are rescuing their function by having UBE3A expressed. So the mice are improving in their symptoms, which we'll talk a little bit more about. And I'm going to show you some videos. You can see on the bottom right that you have all of these changes that are happening where there's no expression with Angelman, there's normal expression with wild type, and we have gorgeous expression with the gene-activated cells. And so these are all things that we have to test in mice to understand how they could apply to humans. And this space, this preclinical work, takes a lot of time, precision, and money. And this is what we've been spending the last 10 to 15 years really doing, and it's all coming to fruition right now. This is a mouse with Angelman syndrome on the left. Looks a little bit like Quincy. Can't keep up, on a treadmill, hold it on for dear life, but a little uncoordinated and really can't keep up. This is after hematopoietic stem cell gene replacement therapy. Same mouse, pre and post. I'm going to highlight a few things here. Firstly, the mouse looks amazing. One mouse, you could tell the difference. Secondly, this was an adult mouse, the equivalent of a 20-year-old, treated, full rescue. Those are the things that are so important for us to remember. They're only mice, they're not people. But that's the best we have in order for allow us to do the most important experiment of all, which is that of people. We have to be safe and we have to understand how it performs. So let's move to pillar 2, but you get the idea. Pillar 2, we're gonna knock down that silencing mechanism. We're gonna hear a lot of talks over the next 2 days about this. We could do that in many different ways, like I said, ASO, CRISPR, zinc finger, miRNA. There's so many ways that we can do this. Unsilencing. This is the data. How does that look? Well, Dr. Art Beaudet, who's in the room and he's going to be speaking after me, he's the first person to have looked at this to say, here's an antisense oligonucleotide, a novel idea of a way we can knock this down and unsilence it. And what you can see from his early work is that we go from no red to red. That means there's no UBE3 in that mouse brain, and now there's a lot. And what he showed is that there's some behaviors that you can rescue in an adult mouse with Angelman syndrome after giving them an antisense oligonucleotide. And more work has been done since that time in order to show that there is potential to make a huge difference in the lives of mice, and now we'll hear tomorrow about humans, with an antisense oligonucleotide by unsilencing the paternal allele. This is zinc finger, same thing. You could see in the middle, AS untreated, it's not very brown. Top and bottom, top is wild type, the normal mouse, bottom is the Angelman mouse after treatment. The entire brain is brown. That's really great. That means that we've turned on UBE3A throughout the entire mouse brain. And here's the behavior, just showing how rigorous this work is and how we're improving the symptoms in the mice with Angelman. But nothing's going to speak to us more than some videos. The same thing has been able to happen with CRISPR, a program in CRISPR at the University of North Carolina, Chapel Hill, out of Yale University, and there's many other CRISPR programs, University of California, Davis, that are working on CRISPR programs. Wild-type mouse on the left, Angelman untreated mouse in the middle. Angelman post-CRISPR gene editing on the right. This is out of Yale University. I might think that our Angelman mouse is doing better than the wild-type mouse, if I may say so myself. Again, this was an adult mouse with Angelman syndrome, treated with a CRISPR gene editor that rescued fully to wild-type or typical levels. And so again, another example of how you can improve the behavior and the function of an adult mouse with Angelman syndrome profoundly. And that's really important to realize all of the strategies and all of the opportunities that we have for our kiddos. Pillar 3, downstream targeting. Let's get in at that synapse, right? The synaptopathy, all the communication between the neurons. What can we do to address that? Well, there's work being done for IGF-1, brain-derived neurotrophic factor. Dr. Marshall's going to talk to us about that today. Oligodendroglial cells, understanding how we can maybe improve the myelination of neurons to make them function better. Dr. Wang is going to talk to us about that today. Alogobat, which is an a GABA agonist, which is going to ultimately be able to help maybe the deletion genotype where we can improve GABA function at the level of the synapse. And so these are all things Roche is going to talk to us about tomorrow. And so we're going to talk a lot about this, and I want you to put it all in context. What category do these talks belong in? Pillar 1, Pillar 2, or Pillar 3? It helps us digest and understand the mechanism of how these drugs might work. Okay, so this is mice. And what I'm going to show you is in gray are all of the behaviors that are tested in Angelman syndrome mice, and there is a lot of them. What do they mean for humans? We can only do our best guess to understand brain function in a mouse and how that translates to a human. But some of the assessments are for cognitive learning and memory, some are motor and balance, some are anxiety and behavior, and some are seizures or EEG phenotype, the delta power that we see, those abnormalities we see on our EEGs. There was a study that was done that was published in 2015 that was a reinstatement model where they had a mouse and they can give it a chemotherapy drug called tamoxifen and activate the gene at different time points. And when they did that, they looked at the behavior to say what can be rescued and what can't be rescued. And what you can see is green is full rescue of the behavior, red is no rescue of the behavior, and light green is partial rescue of the behavior. And what it showed was as you go from prenatal, birth, juvenile, teenager, or adult, you get less and less rescue in the mouse. And so people believe the earlier we treat, the better, and we may not be able to rescue the symptoms later in life like we can earlier in life. And the dogma would say that's true, right? Neurodevelopment, the brain is developing in utero. When you're born, you still have a lot of brain development, but brain development does peter out over time, though neuroplasticity can certainly last into your 30s and 40s, and people are actually saying it lasts into your 50s and 60s. So over time, your learning and memory may still have potential. Looks like seizures may still have some potential, but the earlier time points, maybe not. And that was certainly devastating for parents with children living with Angelman syndrome today when we saw this information. But then we looked at all of the therapies in the mouse model of Angelman syndrome. The true therapy is not a chemotherapy that's inducing an expression, right? And science always contradicts itself. That's science. And you have to replicate and you have to use the same model to compare the same results. But this is using the same model to compare results, not the reinstatement model. That was a different model. And what you can see here is that if you look at the ASOs, the CRISPR, the zinc finger, the gene therapy, AAV, or the gene therapy, HSC, there's a lot of green. I'm going to blow this up a little bit more. Pillar 1, AAV, HSC, okay, gene replacement therapy. Whether you're an adult, you're at birth, you're a juvenile, or you're a teenager, there's a lot of green. So maybe mice don't give us all the answers, right? And really the only answers we're gonna get is in the human population. But this tells me maybe age doesn't matter as much as we thought it does, like I showed you in those videos. Let's look at the ASO, pillar 2, CRISPR, pillar 2, and zinc finger. A lot of green. You can see whether it's birth, juvenile, or adult. There's more green than that first image. And so mice can only give us a certain amount of information. We're going to talk a lot about mice work today, but what I want you to realize is it gives us enough information to realize the possibilities. And the possibilities today are that all of these therapies could have potential impact. And that's phenomenal because most rare diseases do not have these options. We, because of the elegance of the genetics, thanks to the work of Dr. Baudet, who learned and taught us about the silencing phenomenon, we can unsilence or replace or upregulate. Most rare diseases with single gene disorders have one option: replace. We have 13 options, and options equals potential. And that for me is amazing. And so this heat map is something we should be very proud of. Not only because our kids have potential, but because you guys paid for most of this. This is where your money went. It's all coming to fruition now, This is not inexpensive. But you paid for this. This is what FAST does. This is our mission, to figure this out and now get it to humans. So let's talk about getting there. Pillar 4. How do we make sure we advance to clinical trials? There's tons of efforts we have in order to get to trials, and I'm going to talk about all of them. How about our tools? Our tools to get to trials. We've created models. The Angelman Syndrome rat model, pig model, multiple mouse models. Tons of models. We're done with models. Don't come to us with any more grants for models. We're done with models. How about neurons? Well, you guys all supported a biorepository. At Yale University, they're going to talk about this today, we have a biorepository of all genotypes, 5 of all genotypes, and neurotypical siblings, in order to be able to test drugs, whether you're a deletion, a mutation, a gain of function, a loss of function, a mosaic, UPD, ICD. People can test your cells in their drug development efforts, and we've made them freely available to all to have. Because nobody should hold these tools proprietary. We need to share in order to advance drug development for every industry member or every academic institution that wants them. And this is because of you guys. You volunteered your kids' blood to do this. And it is being used every single day. And we can't thank you enough for that. With those cells, we've made landing pads where we can put genes in and out to understand in the deletion, in UPD and ICD with the double silencing, what those genes that are overexpressed or underexpressed are actually doing, and which of those genes we need to target. Organoids, those are mini little brains. From the neurons, they can make 3D structures and test those little mini brains. And Dr. Albert Kyung at North Carolina State University is doing a tremendous amount of work with organoids, and Yale University is doing work with organoids as well, and they're going to share that data with you today. And then we made a human reporter cell line where we can show with a fluorescent protein in a human cell line that we can knock down the antisense and upregulate UBE3A. This has been available in the mouse model, but this hasn't been available in the human cells. And now we have 2 GFP reporter lines, which are human cells that are showing activation. And we've created a bioresource hub on our website that we welcome all of you to go in and look at, where you can see every model that's out there, how to get it, where to get it, what information you need, understanding there's a huge amount of resources that your dollars have paid for, that nobody should be replicating, because it is a waste of your dollars for someone to ask for more dollars of things that have been done. But if we don't know where it is, and it's not publicly available, what happens is people waste money and replicate. And that's not what we want to do. We want to make everything available for all. We created a literature repository as well. Why? Because we have a sophisticated community that loves science and wants to read and wants to understand. And when you read and you understand, you have power. And so we made this super cool repository on our website, the same thing where the literature, where the tools are, where you can go in and you can click on various different things, and it moves and adjusts to whatever you're searching for. And it's very active, interactive platform where you can search thousands and thousands of Angelman articles. And it comes up where the article is, what the abstract is, and if you can have access for free to that article or not, and if you can't, where you can get it. So it's really cool. And remember, the more you read, the more you learn, the more you're going to understand, and the more power you're going to have for your child. So we're very, very proud of the fact that we're creating a resource for you to learn, because it's just so important. The Angelman Syndrome Natural History Study is a phenomenon. This is something that FAST is not responsible for, but we are so proud of the natural history team at Boston Children's and Rady Children's, who really worked so hard since 2006, and there's many, many institutions that have done this, but really Boston Children's and Rady's led the charge. And your kids have been participating in this since 2006. And now we've added FAST Spain, FAST Latin America, and UK and Belgium to the natural history study. And what we can do is we can assess these kids over time to understand what they look like, so we can compare what a therapy looks like and how it might be able to get better. So this is 18 years of data, over 600 patients, 587 as of 2 weeks ago in the US and Canada alone, and over 60 or 70 in Europe and the UK. What you can see is that compared to neurotypicals, which is the gray bar, that gray slope, kiddos with Angelman syndrome are pretty flat. They don't degenerate. They don't go down. They just don't go up. They have a trajectory that goes up until about age 2 to 4, and by age about 5 to 8, regardless of your genotype, you're pretty flat. And a 17-year-old has the same skills generally that are captured on these scales as a 5-year-old, as a 4-year-old. And not to say that they're cognitively a 5-year-old or a 4-year-old, I'm not saying that at all. What I'm saying is that on the scales, you can't measure change because their change and their incremental improvements is very granular, and they're not caught on these measures, which leads us to have to create measures that are better. But it tells us how flat they are and why that's important. So we then launched the Angelman Syndrome Biomarker and Outcome Measure Consortium in 2016, a collaboration between FAST, the Angelman Syndrome Foundation, industry, and academic clinicians, in order to be able to understand how do we measure change in Angelman syndrome. And in 2016, it started with 4 steering committee members and 25 participants in a tiny little conference room. I remember it all so well, thinking about how do we measure change in this population. Well, yesterday, we had over 300 people in a room and online, 25 steering committee members, 11 pharmaceutical companies, over 25 academic institutions involved, 4 advocacy groups, all coming together, 17 talks, including the FDA, and the EMA was present as well. And we'll talk more about that. But this is what progress looks like. And that may not be visible to you, but what we did was we took everything being done in the natural history study, all of the things that were meaningful to our kids and to our families, and narrowed it down on how we're going to measure that. And if we don't have a good way to measure it, we have to develop a tool to measure it. This is all the work that's been done since 2016, just that FAST has funded. $4.5 million. It is so expensive, it takes so much time, but this is also where your money's going. And I want you to realize we're working really hard doing this, but sometimes it's not as visible as a mouse that goes from white to brown or black to red. We have something called the International Angelman Syndrome Research Council. This is where we bring together people from around the world, not just in Angelman syndrome. Actually, we prefer people not in Angelman syndrome, that are working in rare neurodevelopmental disorders, genetic therapies, understanding and how to do a gap analysis on Angelman syndrome. What are we missing? What stone is not unturned? What are the gaps and how do we fill those gaps? This is a partnership with the Simons Foundation and FAST, and we bring together teams every year, and we invite other rare disease organizations. We nominate one new gene every year for their foundation to come and for our brain trust to not only advance Angelman syndrome on day 1, but to advance another disorder on day 2. Because the learnings from one disease will always help another. And if we hold this tight to Angelman, we're not having impact on the rest of the world. And if we're gonna be spending this kind of money and doing this kind of work, there's no reason everybody shouldn't benefit from what we're doing. And so just think about the fact that we're working day and night for Angelman, but the impact that has globally on the periphery that has been arborized to other rare disease organizations is also impactful. And we have 9 other rare disease organizations here today that are here just to listen about Angelman because they look at us as a model. Imagine that. I look at everyone else as a model, but they're looking to us because they're not where we are. So we should be really proud of that because it's taken a lot of work to get here, but we've gotten here pretty quickly considering where we started. Ryan's going to talk about advocacy and the unbelievable work that he has done with the collaboration of the Angelman Syndrome Foundation and you, the advocates, the parents who marched Capitol Hill and said, we need change right now for Angelman syndrome. And I'm not gonna talk about what change we got because Ryan is, but we got it, and we got it. Our 3 asks were successful, thanks to Ryan Fisher. So with all of that, every platform can potentially benefit every child, but different children in different ways in different parts of the world of different genotypes. Each modality may lead to a different biodistribution in the brain and a different result. Different families will have different preferences for different modalities. We don't know what's gonna make it, but we need to de-risk every option so that we have all shots on goal for every individual around the world living for Angelman syndrome, because we don't know what's gonna work the best. We only know in mice, and our kids are not big mice. They're not. So, what we also know is the standard for 15,000 drugs developed, 5,000 make it into the early studies before human, for 10 that make it into a trial, for 1 to be approved. The odds are not in our favor. And the time and the cost is exponential for 1 drug to be advanced. And when Quincy was diagnosed 9 years ago, this is what the pipeline looked like. There were 5 academic institutions working on Angelman syndrome, and FAST was funding 3 of them. 2 years later, it went to 15 because of a huge infusion of funds to FAST, who advanced multiple different projects with huge risk for huge reward. And we went all in with a 24-month plan. 3 years ago, we were at 34 programs. And things were growing and blossoming, and it was unbelievable to see the landscape. But very little was in the clinic. A lot of it was early stage. But 3 ASOs went into the clinic, which was very, very exciting. And then this happened in 2022. The stock market decided that biotech was not a great investment in rare disease, not for the fact that the science wasn't great, but the return on investment wasn't great, and they were changing priorities in the landscape in the for-profit sector. And that resulted in the pausing or canceling of 10 programs. And 34 went to 24. And that is the chutes and ladders of drug development. It's a fact. We have to be ready for it. We have to be prepared for it. We have to accept the fact that our priority doesn't have to be their priority. We wake up every morning and go to bed every night looking into the eyes of a child, saying tomorrow's gonna be a better day. They don't. They don't look into the eyes of your child. They're thinking about strategy, and their strategy has a right to change. But ours doesn't. Ours does not have a right to change. And so where are we today? We're at 27 programs, 15 of which are funded by FAST, marching ever so quickly toward clinical trials. We have 4 clinical trials ongoing right now in Angelman syndrome with human clinical data that's going to be talked about tomorrow. Another program that we're going to talk about that got paused. We have 3 programs in IND-enabling studies that we're going to talk about today and tomorrow, and multiple programs that are being advanced that are further behind. Don't forget Pillar 4, because it's important. This is where we were 9 years ago, and this is where we are today. We're moving all of the pieces together at the right pace in order to get there for our kids, in order to reach that pinnacle of success and approval. And so this is bench to bedside. This is what that looks like. From discovery to preclinical, community, understanding where people are, who they are, what's measurable and meaningful to them, accelerating drug development through companies, through pharmaceuticals, through partnerships, and then running clinical trials. And FAST has decided to invest in every one of these buckets so that we have a true bench-side to bedside model. And that's why last year we told you that the NIH gave $40 million to Yale University, Rush University, and FAST to work together to be able to advance one of the first platforms in non-viral delivered CRISPR therapeutics to neurodevelopmental or neurogenetic disorders. And they wanted Angelman syndrome to serve as the model for other diseases behind it. Why did they agree to that? because we are the lowest hanging fruit. We have all the pieces of the bench-side to bedside model to make it easy to prove if this could work. That's what we're all about. That's why FAST is here. So I told you it's expensive and it takes time. One drug can cost over $200 million to develop before an approval. This is why it's expensive, and this is why we're not done yet. FAST is invested in this spot. We don't want to commercialize. We don't want to take it to market. We want to de-risk it in humans so that the data makes it irresistible for responsible people to take it to phase 3 and bring it to humans. But if we don't de-risk it with human data to show if something works, that's when it gets paused, canceled, or shelved, because it's not valuable enough in this market. And so that's why we're here. And because of that, that's why we decided we needed to be bold, and we needed to really jump forward and advance these programs. And so there's numerous potential therapeutic platforms invested in AS. AS is certainly unique with many elegant gene-targeted opportunities, promising clinical trials to date. We need to accelerate all shots on goal, fill all gaps that we have, because traditional drug development is failing rare disease. It is. The communities need to come together and have been doing this themselves now. And that is a new era of drug development, but it's real. And no one's gonna fail this one because we're gonna do it ourselves if we have to. So we continue to dream big, and that's why we decided to launch an accelerator, a true accelerator. And that accelerator is called AS2 Bio. AS2 Bio is here because we aspire to accelerate drug development for all individuals living with Angelman syndrome and do exactly what I showed you, de-risk to phase 1-2. Get human data, ensure it's safe, it's effective, and find a very responsible party to take it forward and take it to patients around the world. That's our goal, bench to bedside. So we've moved from patient advocacy group through venture philanthropy to a disruptive accelerator platform because we didn't have a choice. And you have to do what you have to do when you have a beautiful child like ours that are asking for hope. So the accelerator is really moving toward clinical trial and acceleration with our goals to advance multiple programs. And the experience you heard about with genetics and ultragenics is not part of AS2, but it is what made us realize it's possible. It's what made us realize we're capable. Prior to the establishment of AS2, we started genetics, and we passed that off to a responsible party. Now we have an AAV gene therapy, a CRISPR gene editor, an HSC gene therapy, and other programs that are earlier on in development. And we're going to do that because we have to. And so, like Alana said, I always say things are either possible or impossible, and there's no in between. And so far, things look really possible. And I think you're going to hear about that over the next 2 days in the hugest way as we talk about mouse data and human data. So that's just my reason. She's one of 500,000. But there are thousands of people here and online listening that have their reason. So we're here for all of you because we can't stop until it's done. And we're not done yet. Thanks. So I think we have time for a few questions. For all speakers, we can answer a few questions after our talks, but at the end of the day, we're gonna have a big panel where all speakers are gonna come up on stage, and we're all gonna be able to ask any question we want to the whole panel. But if anyone has a few questions for me right now, because we are not ahead of schedule, I'm happy to answer a couple questions. I can't see, so scream loud if you have a question. Okay.
I don't see any hands.
It's all, you understand it all now? You got it? No question?
You
have one?
Allyson?
Yeah.
I'm gonna take off my glasses.
How are you, Sue?
Good. With AAV or HSC therapies in mice so far, is there a danger of giving the mice too much UBE3A overexpression?
That's a really great question, and I think that's going to come up a lot throughout the day as we think about overexpression and really giving the gene back, right? What we know is that there's another disorder called Dup15q syndrome, which is a duplication of the 15th chromosome, the maternal allele generally, in which there's the UBE3A gene is duplicated, often duplicated 3 or 4 times. But what's also duplicated are all the genes outside of UBE3A. So it's like the deletion segment of 11 genes that are all duplicated. And what has been shown and what we've looked at in the animal models to try to understand the behavior of duplication and are we getting to those levels with gene replacement therapy is that we're not even close. So what happens is that firstly, you probably need 300 or 400 times in order to get to those levels, and we're going to be, I think, lucky if we get to 50% or 60%. But what we also know is that a huge proportion of the phenotype of Dup15 is actually the GABA genes being duplicated as well. And with gene replacement therapy, we're not duplicating any other gene but UBE3A. And so when we look at the animal models and try to compare them to the duplication model, we're not seeing any of the EEG phenotype or the behavior phenotype of Dup15 with gene therapy. And we know that the mouse is always going to give us more gene therapy in the brain because their brain is so small than we're going to get with a human. A mouse is an overestimation of what we're ever going to get with a human. And so our concern for overexpression, for many more reasons than that, but those are just some examples, is pretty low. And we've had conversations with regulators about this with other programs, and they are comfortable with where we are with the understanding. We won't know until we know, but it doesn't seem like it's something we need to be terribly concerned about at this time. Okay, I'm going to pass it off to Dr. Burdett.
We have a question.
One
second. Running over. I'll be right there. Here. Behind you. Here. There we go.
Thank you. Okay, so, and forgive me, this might be really basic. I'm trying to understand the genetics, but I heard you say UBE3A gene and then UBE3A enzyme or protein. So does that mean the UBE3A gene produces UBE3A protein?
Yeah, exactly.
Okay, got
exactly.
it.
So all genes, generally, you could think about it, that genes make proteins, and it's the protein that carries the function of how our body works. So we have a UBE3A gene, and the UBE3A gene makes the UBE3A protein, and it's the protein that actually works. And something that Barb Bayless is going to talk about is a protein replacement therapy, where why don't we just replace the UBE3A protein rather than the gene itself? We could replace just the protein. And so that's another area that we've been working on as well. But that's exactly right.
Awesome. Okay, thank you.
Yeah.
We have one more question right here.
Hi. Just wanted to know if you've done anything in the mouse models where you've stopped giving the ASOs, for example. What happens to the mice? Do they regress back to where they were at the start or not? And how long does that take?
So we haven't done studies in mice. Most of the studies we've done in mice have been with permanent therapies where you can't reverse it. ASOs would be one example of where you could reverse it. Dr. Burdett can actually talk about this because he did this work early on, but if you give a single dose to the mice, what you can see is UBE3A go up, and then it lasts a couple months or a couple weeks, and then over time it comes back down. The phenotype, the behavior, I don't believe you tested to see if you lost the gains of contextual fear conditioning in those studies. So in terms of behavior, they didn't see if those were lost, but you could see the antisense come back on and the gene go back down. And that in mice, it took a couple weeks, about 14, 16 weeks or so.
We have one more question here.
Hi, I'm a mom.
I see.
I have two questions.
I'm a mom too.
My first question is, for your studies, are you focused on a variety of age ranges in terms of AS, or is your primary focus on the younger or the older? And then my next question is, do you guys have any anticipation on studies of caregivers in terms of their needs and things like that, if that makes sense?
Yeah, those are great questions, and we're working on all of it. So there's been many studies actually over the last 2 years on caregiver surveys of caregiver impact, caregiver needs, and caregiver quality of life and burden. And so I think we launched some of those 3 years ago. And so we've had some really great data, and a lot of that is what we talk about at the ABOM level of the impact of caregivers. We have a patient-focused drug development meeting that Ryan's going to talk a little bit about coming up in January, which is meant to give the perspective of caregivers of meaningfulness and change and burden and desire for change. What's the smallest change that would be meaningful to you, right in front of the FDA. And so those are all very important questions that we care a lot about and we've been doing some work on. As far as age is concerned, again, FAST isn't running any clinical trials, just to be clear. I'm here giving you a summary of all the things that are out there. You're going to hear from the people running the trials tomorrow and some that are in early stages of development today. And they'll be able to answer those questions for you. But what I can say from what I know is that what we showed you, right, is that mice, regardless of age, could have benefit. Our kids don't have a degenerative brain. They have a very healthy brain that is just not making the protein to function normally. And so I anticipate that every trial will include all ages eventually. Everybody's going to have a different starting point. Some are going to start in adults and then work their way down to younger children. Some are going to start in younger children, see effect, and then work their way up to adults. And you could see that with the ASO trials now. One company started quite young, one company was kind of in the middle, and then one was very broad, all the way up to age 50. So I think that it's going to be a smattering, but the take-home message is that adults have been waiting the longest, and there's no reason to believe they won't benefit. Therefore, I think most of the upcoming trials are going to be including adults, if not starting in adults. Okay.
We have one more question here in the back. I'm going to translate because it's a Spanish question. So here we have a gentleman called Octavio. He's 43 years old with Angelman syndrome. And the mom would like to know what the upper age limit is for trials.
That's a great question. You're going to hear a lot about that in the coming talks, particularly tomorrow. So the only clinical trial right now that includes adults is Ionis's ASO trial, and they included up to 50, if I recall, and they're going to be launching their phase 3, as is Ultragenyx, and they announced they're going to be including adults as well. They didn't say up to what age. But knowing that Angelman syndrome, again, is not degenerative, I think it's arbitrary, the number, whether it's 50, 55, 45, 35. But I imagine that there's going to be an upper limit at some point, but more based on comorbidities than necessarily based on a number. But that's going to be based on the company, of which I have no insight into their decisions. I just know that Ionis went up to 50.
Thank you.
Sure. Okay, Dr.
Thank you very much. He has done a lot of exercise, and then I have a lot of and he works now. He's working about 20 years in a bank. So he works with boxes, and he arranges boxes, and he doubles those the hospital. What I can try to tell you is that our children with Angelman syndrome, they can do more things than you can imagine. A lot of things, because they are very, very intelligent. They understand everything.
It's true. It's true. And we're only proving that time and time again. Thank you. Okay.